So now that we've done glycolysis, let's go to the next phase, phase two, if you want to call it that. When we took a single glucose molecule and ran it through the reactions of glycolysis, we made a couple ATP, that's valuable. We made a couple NADHs, that's valuable. We haven't seen why yet, but in the next video you'll see why these are valuable molecules. And we were left with two molecules of this small carbohydrate called pyruvate. It's valuable. It's not as valuable as glucose was, but it still has a lot of value left. And if we could find some way to extract more energy from it, that would be a good thing. It would be efficient. Let me show you an analogy that I like to use for this. Let's say I got a fireplace at home and I go out to the hardware store and I buy one of these Duraflame logs. It's one of these sort of fake pieces of firewood, and you can light it on fire and you can't see it on the label, so I wrote it by hand. These things are supposed to burn for 3 hours. So for 3 hours, I could release energy from this thing, release potential energy in the form of light and heat. Well, let's say I buy one of these, I put it in my fireplace, I set it on fire, I burn it for 15 minutes. Well, I haven't nearly tapped into all the potential energy that it has, but I tapped into it a little bit. I got 15 minutes worth of heat and light. So I burn it for 15 minutes, I extinguish it. But I say, gee, there's still a lot of potential here. I'm going to pick up this partially burned Duraflame log, bring it to my neighbor's fireplace, put it in there, and finish burning it until there's nothing left. So 2 hours and 45 minutes more. That gives me my total of 3 hours. In other words, I take what's left of it and burn it to ashes, but in a different place. So why am I using this analogy? Because that's pretty much what's going to happen to that pyruvate. I took glucose, was my starting material. That's my original best fuel. I, quote, burned it a little bit and got a little bit of usefulness out of it here and here. But then I have the leftovers. This is my partially burned Duraflame log. I'm going to take the pyruvate, I'm going to put it in a different place in the cell, and I'm going to oxidize it to death. I'm going to metabolize it down so there's nothing left to metabolize. I'm going to squeeze every last bit of potential energy out of it. But I need a different place. I need a different fireplace for that to happen. I need a different location to do that. I can't continue to do that in the cytoplasm. So I'm going to take my pyruvates and I'm going to import them into the mitochondrion. Remember the mitochondrion from back when we did organelles? I'm going to bring the pyruvate into the mitochondrion and metabolize it further. This process, known as the Krebs cycle, occurs in the matrix of the mitochondrion. Please go back and review the organelles section. If you've forgotten the mitochondrion, there are various chambers and compartments inside of it. One of those chambers is called the matrix, and that's where the following reactions occur. It's also important to note that cells have more than one mitochondrion, so there are many little fireplaces, if you will, many places where this is going to happen inside any one cell. So during this set of chemical reactions, carbohydrate molecules, specifically pyruvate, are going to be further oxidized. You're going to take pyruvate, which has some hydrogens attached to it. Here's pyruvate. There's a couple hydrogens on it. And you're going to steal all the rest of the reducing power from it until there's no hydrogens at all left. You're going to hand those hydrogens to coenzymes that can make some good use of them. Those coenzymes are NAD and FAD. And when you hand a hydrogen to NAD, you get NADH. And when you hand two hydrogens to FAD, you get FADH2. Please review the previous 2 videos ago when we discussed coenzymes. So most importantly, what you do in the Krebs cycle is you steal the remaining hydrogen atoms from the carbohydrates involved, starting with pyruvate, and you give them to coenzymes that are going to make use of them in the next phase. So this is now a very valuable molecule because it's reduced. This wasn't. This is not a valuable molecule, but this is because it's reduced. So it's the coenzymes that are now holding a lot of energy, and you make a bunch of these and a bunch of these in the process of the Krebs cycle. Also, as a sort of a throw-in, During one of the reactions of the Krebs cycle, you're going to break a chemical bond such that you release enough energy to power the production of an ATP molecule. So you're going to make a little bit of ATP. But what's the waste product of all this? What's the final product? If I take a carbohydrate, carbohydrates are made of carbon, hydrogen, and oxygen, right? If I steal every single hydrogen from a carbohydrate, which is exactly what I'm doing during the process of the Krebs cycle, I'm left with stuff that's made of just carbons and oxygens and no hydrogens. What's a convenient molecule to package up carbons and oxygens and discard? What's a common molecule in nature that's made of carbon and oxygen, no hydrogens, that organisms often discard? Well, carbon dioxide. So a large percentage, not all, but a large percentage of the carbon dioxide that you're getting rid of right now when you exhale through your lungs is produced during the Krebs cycle in the mitochondrial matrices of your cells. So lots and lots of cells with lots and lots of mitochondria in each one, lots of Krebs cycle going on in your body right now. That's a lot of CO2 production. This is as oxidized as you'll ever get a carbon atom. This has no useful reducing power whatsoever. There's no hydrogens attached to this thing. So you throw it away. You excrete it, or you toss it out. For humans, that means primarily through our respiratory system. Other organisms may do it differently. So that's it's a product of the Krebs cycle, but it's a waste product if you want to think of it that way. So by the time all is said and done, for every 2 pyruvates that you start with in the Krebs cycle, you're going to make 2 ATPs. Not bad, a couple ATP. You're going to make 8 NADHs. You're going to make 2 FADH2s. And these are going to be incredibly useful in the next set of reactions in phase 3 of metabolism, so hold that thought. And then your waste product here is going to be some carbon dioxide, specifically 6 molecules of carbon dioxide. Now, I'm not concerned that you memorize how many of each one is produced, but the point is you make a lot of reduced coenzymes in the Krebs cycle. What do you do with them? You're going to cache them in for ATP in the next phase, in what's called the electron transport chain, which is coming up in the next video. You also make a little ATP, that's handy. And then your waste product is this stuff. Not a terribly toxic molecule, so this is a relatively clean burning process. If you would like to see the whole thing, I will attach this image to the video. You can see all the individual reactions, pyruvate, and you can see some carbon dioxides coming off here. Do they illustrate? They don't illustrate. Yes, they do. There's some carbon dioxides coming off of some of these reactions here and here, etc. So those are the actual reactions of the Krebs cycle, which I'm not asking you to memorize, certainly. I just want you to know, again, where does the Krebs cycle happen? What do you start with? Pyruvate. What do you get out of it? Well, you get some waste product, and you get these useful things. What are these reduced coenzymes going to do for you? That's what we're going to look at in the next video.